Hello everyone, welcome to Chemistry Atlas. Today in this video we'll be talking about hemerythrin and hemocyanin. These two topics are very important from the CSR and GATE examination point of view because often question comes from this topic and generally carries a 4 mark or 2 mark. If we can remember all the points related to the structure of both the hemocyanin and hemerythrin, we can easily answer the questions. So let's begin with our today's video. First we'll be talking about hemerythrin. So this is a metalloprotein. It is a non-heme metalloprotein which is responsible for the transport of oxygen in the marine invertebrates. So it is a non-heme protein. So that means there is no heme group in hemerythrin. Talking about the structure of hemerythrin, there are two ferrous ion centers in high spin oxidation state bridged by a hydroxyl group. This hydroxyl group not only serves as a bridging group but also functions as a proton donor to the oxygen molecule when it gets bind to it. Next, one of the ferrous ion center is pentacoordinated and the other is hexacoordinated. Both these iron centers are bound to the protein through the carboxylate side chains of a glutamic acid, aspartic acid, and 5 imidazole rings of the histidine residues. So let us now draw the structure of hemerythrin. We have two Fe2+ centers. They are bridged by a hydroxyl group and one of the iron centers is pentacoordinated aspartic acid, glutamic acid, and these are the histidine residues. So we can see that this center is hexa coordinated and this center is penta coordinated. So when oxygen binds to hemerythrin, this is free hemerythrin, or you can say this is the deoxy form of hemerythrin. So when oxygen binds to this hemerythrin, this penta coordinated iron center binds this oxygen molecule and it transforms to a hexa coordinated center, and both the iron centers oxidizes to ferric ions due to the oxidation caused by this oxygen molecule. So it is Fe3 plus, there will be histidine. I am not writing this part, this will be as similar to this. So these are the histidine groups. The oxygen molecule will be coordinated in such a way, and this bridging hydroxyl group will transfer its proton to this superoxide species, and it will be forming a hydroperoxide complex, and there will be hydrogen bond between this oxygen and hydrogen atom. So free hemerythrin is colorless, and when it gets oxygenated, this is the oxyform. So when it gets oxygenated, it becomes violet pink. Here both the iron centers are in high spin oxidation state, and here both the iron centers converts to Fe3 plus, that is ferric ion, and they reside in low spin oxidation state. When oxygen binds with the free hemerythrin center, it gives a Raman stretching frequency around 850 centimeter inverse. This complex will be EPR inactive due to antiferromagnetic coupling between the ferric ions. That is, there will be no unpaired electrons present in the oxyform, and as a result it will be EPR inactive. So these were some important points regarding the properties related to the structure of hemerythrin. These points will be highly beneficial if we can remember them, and these points will definitely help us to answer any of the questions in the examination. So let's move to our next topic, hemocyanin. So hemocyanin are a family of copper containing proteins which are found in non-insect arthropodic groups, acrinids, like the horseshoe crab, octopus, etc. It is also a oxygen transport metalloprotein. Oxyhemocyanin is blue in color, while in the deoxy form it is colorless. Talking about the active site of hemocyanin, it is composed of a pair of copper atoms in +1 oxidation state, which are directly coordinated to the protein through the imidazole rings of 6 histidine residues. So they are connected like this. So when it undergoes oxygenation, the copper centers are oxidized to +2 oxidation state, and there is a eta 2 linkage by the oxygen molecule. This is a eta 2 mu linkage of the oxygen atom. Here eta 2 represents the hapticity of the oxygen atom, that is, one oxygen atom is coordinated to 2 copper atoms. Similarly, this other oxygen is also coordinated to 2 oxygen atoms, and there is a mu linkage, that means it is forming a bridge between the 2 copper ions. This oxyhemocyanin gives a Raman stretching frequency around 750 centimeter inverse. In the oxy form, the hemocyanin is EPR inactive due to the antiferromagnetic coupling of the two Cu2 positive ions. So, comparing the structure of hemocyanin and hemerythrin, the main difference can be pointed out is that in hemocyanin, the two oxygen atoms forms a bridge between the two copper sites, whereas in hemerythrin, oxygen atom is linked to one Fe site only. So these were some important points, properties of hemocyanin. In the next video, we will be taking some questions that came in the previous year examinations. So I hope you like this video. Feel free to subscribe to the channel. Thank you.